finally get that recording up. I had trouble getting it posted from home, and I learn a little bit every time. So hopefully that will go more smoothly. Remember, next time, I posted an announcement on this, next time you'll need to come in the door, like on this side of the building, and then we'll be all exiting the door and then coming in. So have your ID ready. Apparently they're going to make everybody scan it because the rec center will be open. So one more thing to kind of get used to here. All right, so I want to start throwing in some old test questions every once in a while. Not really doing clicker type questions, but at least you get to see a few questions that I've asked in the past. This semester, we don't really talk very much about viruses yet, and I know that feedback is a little bit annoying. Always let me know if you can't hear me or anything. I can't do a lot about that. Try to minimize it a little bit. Do this. Okay. So viruses not included in that taxonomic scheme. So last time the last couple times we've been talking about Carl Woese, and remember how he sequenced, well, you have to know a couple things to answer this question correctly, but he sequenced ribosomal RNA to figure out that phylogeny, those 3 different domains of all organisms. Okay, why aren't viruses on that phylogenetic tree? Well, they lack ribosomal RNA. They don't have ribosomes. So it's a little bit of a trick question. So it's one of the reasons that I like to challenge you with little harder questions in class and then try to leave some of the easier ones for the exam. But our exam is not next week, but the week after. So kind of on the horizon there. A lot of you have been starting your homework, some of you not. I know that's sort of a personality thing, but I would encourage you to kind of jump in there and give that a try. If you are having trouble still with mastering or you don't have that purchased yet, talk to me about that. I've set back that due date of that first little couple question assignment for a few people. So again, that was just sort of encourage you to get started on things. So don't be afraid to talk to me about that. All right, so I know last time probably, I didn't have anybody kind of tell me I was going too fast last time. I know what I had up on the board, you were probably hopefully trying to scribble down a little bit. I did record the lecture the way that I meant to give it in class. So I think the content is almost exactly the same, but there might have been a couple variations. Don't be afraid to ask me if you have questions about that. But we ended last time talking about that acid-fast cell wall. Of course, we talked about what's special about gram-positive cell walls, what's special about gram-negative cell walls, the different structures we see there. And then we have this other class. It's almost like an exception to those to that rule. We have this class of organisms, they're mycobacterium, they're considered acid-fast. They still have peptidoglycan in their cell walls, but they have a lot of other stuff there too, in particular mycolic acid, which gives the cell walls a really waxy kind of covering. And I wanted to talk a little bit about how we stain cells. Remember, these are the mycobacterium. And in lab, typically we would be doing that stain here in the next week or two. It's one of the things we kind of just deemed non-essential. So I want to talk a little bit more about that staining procedure. Whenever you're staining cells, you typically mix them in a drop of water. If you're getting your cells from some kind of solid media, so you make a smear of cells. And typically you run them over the Bunsen burner a few times, or you put them on a hot plate real low to heat fix them. So we try to get our cells to adhere to the slide a little bit so they don't just wash off when we start applying stain. That's typically the way we do it, not every time, but typically what we do. And then when we're acid-fast staining, our first stain that we use is called carbylfusion. Kind of a tough word to spell. Sometimes this is called Kinyoun stain. But you might be thinking, well, why don't we just stain these cells like any other kind of cells? Well, the dyes that we use when we're gram staining, for instance, they're dissolved in water. When we talked about this waxy cell wall that the mycobacterium have, those gram stains just don't adhere very well to this waxy cell wall. Carbylfusion, what's special about this dye is that it's dissolved in phenol. Okay, so that it penetrates into these acid-fast cell walls. And we leave the dye sit on there for a good long time, usually about 10 minutes. And then we rinse it off, and then we add a decolorizer. It's acid dissolved in alcohol. Actually, hydrochloric acid. Okay, so this is where the name comes from, acid fast. If an organism has this specialized kind of cell wall, it holds fast to this carbylfusion even when it's washed in acid. Okay, so not quick fast, but holding fast. So holds fast to carbylfusion. So our acid fast organisms are these hot pink ones here. They're kind of clustered together. They're actually rod-shaped organisms, bacilli, but they tend to kind of stick together. And this carbylfusion is a hot pink color. All right, so our acid-fast ones are hot pink. Any organism that doesn't have an acid-fast cell wall gets decolorized. So we have to add another stain so that we can see them at all. And we typically add methylene blue because that's a nice blue color contrast from the hot pink. So anything that's not acid-fast, like these staphylococcus here, are going to stain blue. All right, so that's how you would do an acid-fast stain. Yeah.
So is it the metallic acid or is still the peptidoglycan that holds on to that primary stain for the coloration?
Okay, so the question was, is it the mycolic acid that holds on to the primary stain, or is it the peptidoglycan? Probably a mixture of both. I think of course, the mycolic acid has a lot to do with it, why we use that special stain, but good question. I don't know exactly how to answer that, but good. Okay. All right. So moving on here. So we've spent a lot of time, remember, we're working into our bacterial cell from the outside on the way in. All right, so after the cell wall, we come to the cell membrane. And I'm not going to spend a lot of time here because this should be, the structure of this should be fairly review for you. The membranes of bacteria are very similar to our membranes. Okay, you should recognize the fluid mosaic model here. Functions of that semi-permeable membrane is to let some things in and some things not. So it controls passage of things in and out of the cell. We'll be talking more about this when we get to metabolism coming up here in the next few weeks. Basically, it has to maintain a concentration gradient, maintain that electron gradient, and that leads to the production of ATP during aerobic respiration. So we'll see that. So this is where lots of the enzymes that are necessary for that electron transfer chain are going to be located in the membrane of prokaryotic cells. Of course, eukaryotes have some specialized structures like mitochondria there, but prokaryotes, again, have a lot of that in the membrane. Okay, so let's talk a little bit about transport. Again, a little bit of review here. Of course, we're going to talk first about passive processes. So that means that no ATP is required for these kinds of transports through the membrane. Obviously, diffusion, it's just high concentration of some substance will, without energy, flow into areas where there's a lower concentration of the substance. So some things can diffuse right through the membrane. Things like water, carbon dioxide, oxygen, simple sugars, okay, they can usually easily diffuse through the membrane. Sometimes a protein channel is necessary for things to come through the membrane, and that can either be a non-specific channel, we call that facilitated diffusion when a protein is necessary, or it can be a very specific protein to let a specific thing into the cell. And in that case, we usually call it a permease enzyme. For instance, lactose needs a permease enzyme to bring it inside of the cell. And then, of course, when water moves from areas of high concentration to low concentration, we call that osmosis. And sometimes that can happen through a protein, sometimes that can happen without one. All right, so a little review of osmosis. It's one of those concepts that you've seen over and over again, but I still often see students a little bit confused about that, and it is one of the kind of big things in biology that you're just going to see over and over again. So I just want to review the process of osmosis, which way the water moves. Let's look at it first, how that affects a bacterial cell. So if a bacterial cell is in an isotonic solution, of course, these terms, hypertonic, hypotonic, isotonic, refer to the solution that the cell is in. So you never would say, oh, the cell is hypertonic. Okay, you always did describe the medium in relationship to this cell. Okay, so they're, again, relative terms. Something's not always hypotonic, it's hypotonic compared to what? Okay, and again, we use those to describe the solution because the solution that the cell is in is likely to change much more quickly than the actual chemistry of the cell itself. Okay, so that's one thing that is easy to get goofed up. Of course, if it's an isotonic solution, the net movement of water is really the same in and out of the cell. A hypertonic, that means high solutes outside of the cell. So if I put something in brackets, that means concentration. So high concentration of solutes outside of the cell. Okay, so if that's the case, then you have a lower concentration of water outside of the cell. And water moves from high concentration to low concentration, right? So we're gonna have water leaving the cell in a hypertonic medium. And you can memorize that, but I think it's helpful to kind of be able to think through it as well, why that's the case. Okay, and then hypotonic, lower solutes outside of the cell. Again, some solute that can't easily go in and out of the membrane. In that case, we've got water. Water follows stuff. That's what a student told me once, okay? So same thing, we've got, again, low solute concentration outside of the cell. So the water concentration is going to be higher, and it's going to move from high concentration to low concentration. So notice what's happening to the cell here. When the cell is losing water, we see the cell membrane kind of crenating a little bit, but the cell shape pretty much stays similar. This is a very bad situation, a hypertonic situation for a cell to be in because it's losing water, it's dehydrating. And that's bad for bacteria cells. They like to stay moist. So a hypotonic solution where we have water entering the cell doesn't mind that too much. It's got that nice, strong cell wall to make it able to withstand that. So it would take a huge gradient in that solute concentration there for that cell to burst. Unlike an animal cell or one with no cell wall, okay, that's very easily going to burst if you have it in too hypotonic a solution. Same thing though with the bacterial cell. If that cell wall is got some holes in it, or some of those amino acid cross bridges are not formed, then this cell is going to be susceptible to bursting due to that hypotonic solution. Okay. All right, other means of transport. Okay, here now we have active processes. So that means it requires the cell to spend some ATP. So usually there's some kind of carrier proteins that are going to be utilized here. And I'm not really too concerned about these different terms of that there, but if a uniport, that just means, you know, one substance is brought into the cell, ATP spent. An antiport, one molecule goes one way, one the other. Usually it's going to be some ion that's allowed to diffuse out and then to spend some energy. This just makes it a little bit more energy efficient somehow, spend less energy when one thing goes one way and one the other. There can be a couple different proteins that are coupled together. And again, usually one thing is allowed to diffuse out and then, you know, energetically, it just makes it a little bit more favorable to bring something else in. There is one kind of transport. This is unique to prokaryotes. And again, this is an active process, so energy is spent here. And this is really a really clever way to bring something into the cell against the concentration gradient, and then to make sure that it doesn't just diffuse right back out again. Okay, so the best example of this is to use some energy to bring glucose into the cell. And during group translocation, it's chemically modified. So phosphate is added to that glucose. So now we have glucose 6-phosphate. And you may recognize that that's the first step in glycolysis. Okay, so the glucose, a phosphate is added to that. And now the glucose won't just diffuse right back out again. Okay, it's chemically changed. And so it's not going to flow maybe back down the concentration gradient. All right, so again, that's only prokaryotes that can do that. And we're going to talk about some means of transport that only eukaryotes can do as well. All right, so moving into the cell. Okay, we spent a lot more time outside of the cell actually than inside of the cell. What's in the cell? Cytoplasm and cytosol. Until maybe a few weeks ago, I used those words interchangeably, but actually there is a slight difference in the meaning of those two terms. The cytoplasm is basically anything that's contained by the cell membrane. So like right down here in this little cell that I have pictured, the DNA, it's in the cytoplasm. Okay, the cytosol though, this is actually the term, it's the fluid present inside the cell. Okay, so this is mostly going to be water. There's also maybe some ions in there, some small proteins. Okay, so basically the cytosol makes up the cytoplasm, or the cytosol is in the cytoplasm. Okay, again, those terms are often used kind of interchangeably, but there is technically a slight difference there in what they're meaning. Okay, well, what else do we see in the cytoplasm? We find the cell's DNA. Okay, and that's going to be located in a region of the cell called the nucleoid. Okay, so it doesn't really show that in this little picture I have here, but of course, remember, there's not a membrane around the DNA. It's just still kind of floating in, we call it a region of the cytoplasm called the nucleoid region. And so we'll talk more about what form that DNA takes in prokaryotic cells when we get to our chapter on bacterial genetics. But Usually organisms are going to have a chromosome, and bacteria often have one, maybe two. Okay, there's all sorts of weird exceptions there, but usually they have one or two, and usually it's going to be circular. Again, there's some exceptions to that. There's some organisms with linear DNA, but it's usually going to be like this, a great big circle of DNA. In addition, they often have multiple plasmids, usually, again, lots of them. These are circular and smaller pieces of DNA. Okay, and typically the plasmids are going to encode for something that the cell, that's not absolutely necessary for the cell to survive. Okay, so like the enzymes for glycolysis, those are going to be on the chromosome. But plasmids might carry information like antibiotic resistance or other things that might make the cell a little bit more suited to live in certain conditions, but not absolutely necessary to live anywhere. Okay, so we'll talk a little bit more about that. And there's not really a whole lot else inside that cell. Because there's no membrane-bound organelles, there's not a lot else, but the cell does have ribosomes. So we'll talk about that. They're not really membrane-bound. That's why anyway. Okay, so you should be familiar what ribosomes do. That's the site where proteins are synthesized in a cell. And I thought we'd go ahead and look at the structure of both prokaryotic ribosomes as well as compare them to our eukaryotic ribosomes. Similar structure, not exactly the same. So there's always going to be two subunits. And those subunits are made of ribosomal RNA and proteins. So you can see down here, for instance, in this large subunit of prokaryotic ribosomes, there's a little red line there that is supposed to be the ribosomal RNA. It's called 23S. Okay. S is a unit of sedimentation. S means Svedbergs. Okay, and that's, again, a unit of sedimentation rate. Okay, so for example, that large subunit has a 23S piece of ribosomal RNA and a 5S piece. Okay, but then it adds up to, I can't even read this, 50S. Okay, so what kind of addition is that? Well, the sedimentation rate is a function of the mass and the weight and the shape of that ribosomal RNA, so it doesn't necessarily add up there. You got a bunch of proteins mixed in there that make it sediment a little bit slower. Okay, so prokaryotes have, when the small unit and small subunit and the large subunit come together around the messenger RNA to actually start translating proteins, they come together to this 70S ribosomal complex. Okay, eukaryotes, a little bit different, 60S and 40S come together to that 80S. Okay, so pretty similar structures, different enough that we have antibiotics that target prokaryotic ribosomes and not eukaryotic ribosomes. So that's certainly a difference that we exploit. Okay. And the archaea have ribosomes that are pretty similar to the prokaryotic ones, same size, 70S. The shape is a little bit different of the archaeal ones. Okay. All right, forget to say anything there. All right, so other internal structures that are present on some bacteria and not others. So we talked briefly about endospores last time as a way to classify organisms, whether they're able to make endospores or not. Let's talk a little bit more about this structure. Remember, this is a survival mechanism for bacteria. It's not reproductive. If you'll see in this little sporulation life cycle that I've got here, the vegetative cell here, this just means the cell is metabolically active. So if you've got a metabolically active cell and it encounters some kind of, let's say, harsh environment, that might cause it to start to sporulate. And you can see we end up with one free endospore from one vegetative cell. So that's not reproductive. As we'll see when we look at fungi, they make spores as well. They make hundreds of spores from one structure. So that's a reproductive structure. So more and more we're moving to calling, we used to call this a free spore. More and more we refer to it as a free endospore. We used to call it endospore when it was still inside the vegetative cell. But if we call it an endospore, then it helps us differentiate spore formation in bacteria from that in fungi. Okay, what kind of cells are able to sporulate? What kind of bacterial cells? Here's a couple genera names that would be good to recognize: Clostridium and Bacillus. There are other spore-forming genera in bacteria, but these are the two most famous ones by far. And they cause, the Clostridium especially causes a lot of really interesting infections. We'll get to some of them at the end of the semester. Clostridium difficile, C. diff. If you work in a nursing home, you've heard of that for sure, probably smelled it. Tetanus, botulism, those are all infections caused by Clostridium. So pretty nasty genera, a lot of toxin formation there with that one. Bacillus is a genus of bacteria that's pretty harmless for the most part, soil organisms. Some of them make antibiotics, but there's one notable exception, and that's Bacillus anthracis, causative agent of anthrax. So some pretty interesting infections that are caused by spore formers. Again, this is very rare in the bacterial world. There's 20-some genes that are all necessary for the cell to be able to go through this spore forming process. And incidentally, these are both gram-positive organisms, big gram-positive rods. So what kinds of harsh environments can cause a vegetative cell to start sporulating? An ideal environment for most bacteria is a nice, warm, moist place. So anything different than that. So maybe dry conditions, certainly low nutrients can cause sporulation. Temperature extremes, especially high temperatures. You can probably think of a lot of other things like extreme pH, exposure to radiation like UV light. But just keep in mind when a cell encounters those environments, it takes somewhere on the order of 6 to 8 hours for it to go through that spore forming process and end up as a free endospore. Okay, so we think that some of these cells are just normally going through this spore cycle just so that there's some spores in that medium or population of cells all the time. Okay. And this spore coat, we won't really get into talking about why these endospores are so tough, but this spore coat, lots and lots of layers there. The DNA is dehydrated a little bit. These free endospores basically can stay viable We don't even know the upper ends. Certainly decades, centuries. We found spores in Egyptian mummies that are thousands of years old that have still been able to germinate. So extremely tough structure, hard to kill, not impossible. Okay, we can heat it up enough to get rid of it. We've got chemicals that can kill spores, but pretty difficult to kill.
So does it only form a spore when it's in a harsh environment?
So good question. Does it only form a spore when it's in a harsh environment? It's more likely to, but we think that some of them are going to go. So in lab, we usually, to answer that question, we usually treat them with low nutrients to try to see a lot of spores so that we can see them in lab. But it makes sense for there to always be a few endospores in a population of cells, because if I were to suddenly, you know, heat that up for like 10 minutes to whatever, 99 degrees Celsius, all the vegetative cells would be killed. But if there were a few spores in that medium, then they would be able to germinate and replace themselves. So if the conditions change really quickly, they want to have a few spores around to be around. Okay, so good. Spores are one of my favorite topics, very, very interesting. I do want to talk a little bit about the spore stain, again, because most of you will not get a chance to do this in lab this semester. There are several different acid-fast stains or way to do it, or at least one other major one, but at least with the spore stain, the Schaeffer-Fulton method is kind of, as far as I know, the only way that's typically done. And this is the way the cells will typically look after we do that spore stain. We put our cells on a slide, we heat fix them, like I mentioned before, and then we add a stain called malachite green. And there's nothing really special about that dye, but what we do special to it is that we apply heat to the cells when the stain is on there. We usually put our slide over a water bath that's boiling. So we apply the steam for about 5 minutes, and that disrupts the spore coat enough that to allow it to take up this malachite green. Okay, so these little green ovals that you see, those are endospores, again, that have been stressed enough to take up that malachite green. We just rinse the dye after it's been on there long enough, and then we add a second stain, safranin, which is a red dye. We use that when we're gram staining as well. That stains the vegetative cells. Okay, so for some reason, those metabolically active cells don't take up the malachite green very well, and the cells here, the big rods here, those are the vegetative cells that stain red. Okay, and hopefully, if you know, lucky enough, you get to see a few cells where the spore is still inside of the vegetative cell. Okay, and I just included this picture. This is probably some Bacillus anthracis that have been gram stained. So when we gram stain spores, they don't take up that gram stain dye very well because we haven't heated them. So in this case, you can still barely make out those free endospores, but they're it'd be easy to miss them, especially if you didn't see any cells that had an endospore in them. They basically just don't take up the dye very well. Okay. And anthrax has this really characteristic boxy shaped bacilli that's pretty easy to spot. Okay. All right. So the only other structure that I want to mention that's inside bacterial cell, and again, just some bacteria, are inclusions. And there's a lot of different kinds of inclusions. Certain bacteria will be able to collect certain ones, but these are just storage sites. Okay, and the bacteria could be storing a variety of different things for a time, say when the nutrients are not as available. Could be some kind of lipid or starch or some other necessary chemical. And there's a couple examples of inclusions. One that's particularly interesting is polyhydroxybutyrate. There are certain bacteria that will store, it's kind of a, let's see, it's kind of a starchy polymer, we'll call it. And here's an example. So here, each one of these kind of circles there are some blobs of this PHB. Again, it's a starchy polymer. If you grow bacteria on a certain kind of media, you can get them to hoard up a whole bunch of that, and then you can collect that PHB and alter it just slightly and turn it into a biodegradable plastic. Okay. So let me show you this picture here. Here's one of these PHB bottle made out of PHB plastic, and when it's put in the landfill, basically other bacteria break it down within a matter of weeks. So of course that's not going to be a good solution to make all kinds of plastics out of, you know, things that are going to break down really quickly, but it's kind of cool when you think about how we can use bacteria to do things for us much more cleanly. Okay. Another example of an inclusion are just gas vesicles. Oh. Okay, you know what that is? Okay. A bacteria that is photosynthetic needs to situate itself in the just right place in the water column so it can store up the appropriate amount of, you know, whatever gas and use it as a flotation device. Okay, so that's just a couple examples. There are lots of other ones there. Okay. All right, so still in chapter 3 here, but we're going to move on and think about some eukaryotic structures. I'm going to mostly move through these a little bit more quickly because when we're talking about the organelles, for instance, I'm assuming that you have those in other classes, and I'm really only interested that you have sort of a very basic understanding of what those structures do. As we talk about some of our microscopic eukaryotes at the end of this lecture and into next, really you just need a kind of a basic understanding of that, as well as for when we start talking about immunology. I do want to highlight structures that bacteria and eukaryotes may share, how the structure may be different in eukaryotes. So we talked about how lots and lots of bacteria secrete some kind of glycocalyx, whether it's a capsule or a slime layer. It turns out that eukaryotes can have a glycocalyx as well. But it's always going to be anchored to the cell membrane. Okay, we talked about the prokaryotes, of course, this was outside the cell wall. Okay, so eukaryotes that have a cell wall do not have a glycocalyx. Okay, so not found in eukaryotes with a cell wall. All right, so eukaryotes with a cell wall, things like fungi, plants, algae. Okay. All right. Same composition. It's always going to be some kind of polysaccharide composition. Not always. Never say always. Most of the time. Okay. And it's usually a little bit more loosely defined than what we would find in bacterial cells. Functions are very similar here. Attachments is a big one to, you know, wherever it needs to be. Some protection against drying out. One function for prokaryotes was it protects the cell from being phagocytized. Eukaryotic cells are so big that typically they're not going to be phagocytized by another cell, so that's not a function, but it certainly helps strengthen the cell surface a little bit, especially because these cells don't have a cell wall. And the cell wall, I'm not going to talk a whole lot about here. We spent a lot of time on that for our bacteria. Of course, which eukaryotes have the cell wall? I just mentioned that. It's fungi, plants, and algae. Not animals, of course. Same function as prokaryotes. Strengthen that cell surface. Support, shape. The composition is going to be quite varied. So we're not going to go into a lot of detail there. For plants, they have an awful lot of the polysaccharide cellulose. It's a much simpler structure than what peptidoglycan is. Fungi, cell walls composed primarily of chitin, another fairly simple polysaccharide. Some protozoa, they don't really have a cell wall per se. They have a structure called a pellicle sometimes, which is made of protein. Proteinaceous pellicle. Okay, so not really a cell wall. I'll just kind of mention that there. Okay, but the main thing, remember, the composition is never going to be peptidoglycan. Okay, that's a hallmark of bacterial cells, cell walls. Okay. And again, cell membrane structure very similar to bacteria. One difference is that they have some sterols, in other words, steroid lipids in cell membranes. I should say we have that. So cholesterol is something that we have in our membranes that bacteria don't. We think about cholesterol as being bad, but it's necessary to help keep our membranes fluid in cold environments and give them a little stability in hot environments. Of course, function, semi-permeable barrier, a lot of the same functions as bacteria. One different function that bacteria can't do because they have a cell wall outside their cell membranes. Some organisms can kind of, can't find the word that I'm looking for. They can actually move their cell membranes enough to extend pseudopods. Okay, so let me just, I only posted, I think maybe this part of this figure from your book here, but here's a pseudopod that this amoeba is extending. So it's reorganizing, that's the word I was looking for, reorganizing its cell wall a little bit. There's microtubules at work there that we didn't really talk about, but they're extending a pseudopod, and that's a way that amoebas move around. Some of our cells do that as well. Our white blood cells can do that and move in amoeboid motion. So because of that reorganization that can take place, because there's no cell wall, we see some distinct kinds of transport that we don't see in bacteria. Okay, endocytosis and exocytosis. Okay, so endocytosis is when an organism will extend pseudopods around some kind of substance, and that we could call pinocytosis if it's going to envelop some kind of liquid. And the more familiar term, phagocytosis, is when some kind of solid like some bacteria or smaller organism gets engulfed, basically, and brought into the cell. Okay, so here's, showing some phagocytosis. This amoeba is extending the pseudopods here in order to engulf some smaller organism and use it for a nutrition source. Question in the back. So the question is, didn't I say that eukaryotes couldn't phagocytize because how large they are? If I said that, I misspoke. What I meant is, oh yeah, sorry, right. So you have one protozoa engulfing another protozoa, right? So yeah, I misspoke before. I guess what I meant is that certainly a bacteria wouldn't engulf a eukaryote, but right, you could have a larger eukaryote engulfing a smaller one. So yes, so they can do it to each other. Yes, so a smaller eukaryote, right, can be phagocytized by a larger one. So yes, thanks for, clarifying that. Right, so, but the point is that bacteria can't do this because they have cell walls, all right? So they don't have that liberty to extend their cell walls. And exocytosis is just when the organism is getting rid of some waste, typically. Okay, so we'll see that in action when we see, like, we're gonna talk a little bit further about what happens to whatever is getting phagocytized and how the organism breaks that down. All right, so the next structure that we would come to, working from cell wall, cell membrane, next structure we would come to then is the flagella. So if you remember when we talked about bacteria, we did the flagella before the cell wall because it was an external structure. With eukaryotes, it's actually membrane bound. So any flagella, if they are present, are going to be encircled in that cell membrane. And I should say the flagella, basically the function is the same between prokaryotes and eukaryotes, but that's pretty much the only thing that's the same. Okay, we have a totally different structure for eukaryotic flagella. Okay, they're composed of microtubules, and it's called a 9+2 arrangement. And that has always seemed like a strange name to me. My son, when he was younger, was looking over my shoulder and said, well, that's really dumb. Why don't they just call it 11? He was always good in math. The reason they call that is because you have 9 pairs of microtubules that make up that flagella filament, and there's 2 in the middle. Okay, so that's where the 9+2 comes from. Those microtubules I wanted to mention are made of the protein tubulin. Okay, so remember for bacteria, it was made of flagellin, a different protein. Okay, and down here at where the flagella is going to be anchored inside of the cell, the structure actually changes a little bit. And we have a 9+0 arrangement, okay, down in the basal body. Okay, why that structure changes is kind of a mystery. All right, and remember for the bacteria, we had a filament, hook, and basal body. Eukaryotic flagella, there's not going to be any hook, just the filament and basal body. All right, so like I said, function is the same. They're going to be moving towards some kind of attractant, so they're chemotaxing or phototaxing if it's a photosynthetic organism. The motion is different as well. Remember, the bacteria flagella moved, they rotated either one way or the other. They move for eukaryotes in a whip-like fashion. Okay, and I'll talk about, there's a couple easy ways to remember that. But here's a couple of nice YouTube clips. The first one, this one's all about euglena. And so there's like 10 or 12 minutes of different, you know, euglena buzzing around. One of the cool things that I didn't realize till, you know, I started teaching this class is that a lot of eukaryotes that have a single flagella here, they actually move in this direction towards the flagella. So you see that in this clip, you can just kind of see the flagella wiggling around a little bit and it's moving towards it. You have a question? Oh, so question is, if the only thing that's the same is the function and it's totally different other than that, why do we call it flagella? Maybe we should have a special name for it. I guess they look the same. I don't know. That's a good question. Definitely looks the same, and again, same purpose. It's just the whole structure of it is different, is what I meant. So some with one flagella, a lot of them move towards the flagella, some move the way that I would expect them to move against it. And this is a short video of sperm. And you can really see the motion of the flagella really nicely in that clip. Yeah, question?
So bacterial flagella, filaments and
Flagellin.
What's the difference between microtubules and flagella?
All right. So good question. What's the difference between tubulin of microtubules and flagellin of flagella? I would have to look that up. So that's a great question. Yeah, we're just not going that deeply into the structure. That's a great question, and I'll look it up and see if there's anything particularly interesting there. Maybe they are very similar, but that's a good question. Okay, so the way that makes sense to me that I can remember that the eukaryotic flagella move in a whip-like way actually is when we talk about cilia. Cilia are pretty much the same. It's got the same structure as flagella. They're only actually obviously much shorter, and usually cells have a whole bunch of them surrounding the cell. We have ciliated cells lining our respiratory tract, okay? And they're helping to expel things that don't belong there out of the respiratory tract, okay? So think about, since the cilia are very similar to eukaryotic flagella, it wouldn't make sense for those cilia to be spinning around. That wouldn't be helpful. We want them moving in a whip-like way. So if you remember that, again, eukaryotic flagella, very similar to cilia, then that makes sense that they're moving, again, in a whip-like way. And if all these flagella on a, say, whatever that protozoa is, were spinning around, that wouldn't be able to move it in a purposeful way, I don't think. Okay, so only eukaryotes have cilia. Remember, yeah, I won't mention that. Okay, usually have lots of them, between 100 and 1,000 on the surface of the cell. Movement, circulation of material is going to be the function there. Okay, so mostly protozoa and a few types of animal cells. Okay, the only example I can think of there is our ciliated epithelial cells. Okay, what are we doing here? All right, good. So moving, again, I'm gonna move fairly quickly as we look at these structures inside the eukaryotic cells. Of course, you know, there's a nucleus there. It's the largest organelle. Usually there's only one per cell. There can be lots of exceptions to that if the cell is infected with a virus. Sometimes that makes cells fuse with each other and they become multinucleated. We see that, of course, red blood cells have lost their nucleus. So there's certainly some exceptions there. But of course, it's the control center of the cell and it's where the DNA is located. Okay, it's got a double membrane around it. There is, of course, nucleoplasm. Okay, or cytoplasm in the cell. We've got nucleoplasm in the nucleus there. And this is written too small for me to see, but this is the nucleolus. Okay, that's the part inside the nucleus that contains chromatin. During mitosis, that condenses and becomes visible as chromosomes. And what else was I going to say about that? And the nucleolus is also where ribosomal RNA is synthesized. Once we move outward from the nucleus, we would first come to the endoplasmic reticulum. And of course, there's a rough endoplasmic reticulum, and then out further than that is the smooth endoplasmic reticulum. In general, this is kind of like a big transport system. And of course, what's different between the rough and the smooth? The rough contains ribosomes. Or it's studded with ribosomes, basically. So why are some ribosomes free and some are in association with the ER? This is where proteins are made, proteins synthesized, that will be exported from the cell. Okay, so there's lots of instances where a cell would have making a protein that it wants, again, to be used on other cells rather than its the cell itself. I'll show you an example of that in just a minute. We could spend a lot of time on functionality here. In general, what goes on in the smooth ER, lipid synthesis is one of its big functions. But it's really, again, if you think of it as kind of a transport system, that's a very reasonable thing. I don't think I posted this picture, but it's just striking to me when you learned about the organelles of a eukaryotic cell, you know, whenever you were growing up, you always see a cell that has one nucleus and one ER and one lysosome, and, you know, kind of one of everything, right? That's not typically the way it works in cells in our body. It depends what kind of cell it is as to the proportion of what kinds of organelles are in there. And if you look at this cell, of course, it has a huge nucleus. And then basically the only other things that you can recognize, these little blobs here are mitochondria, and all the rest of this is rough ER. That gives you a hint as to the function of this cell. Its main function is to make lots of proteins that get used elsewhere. They get exported outside of the cell. And plasma cells, we're going to talk about those later on, part of our immunology unit. Those are cells, you know what plasma cells make? They make antibodies. Okay, so a plasma cell is going to be pumping out thousands of antibodies a minute, or a second, sorry. Okay, so very metabolically active, and again, lots of rough ER, so that's not too surprising. The next organelle you would come to then is the Golgi complex. Not all eukaryotes have those, surprisingly, but it's, again, more of kind of a packaging altering a little bit some of those proteins that might be made in the rough ER. Okay, so vesicles kind of pinch off from the smooth ER and then kind of add on to the Golgi there. Modify contents. So in the Golgi, you could take a protein, again, that was made in the rough ER and attach a sugar to it. So now it's a glycoprotein and not just a protein. Okay, you could add a lipid to it, something like that, some kind of modification. And then an organelle that we will spend a little bit more time talking about later are the lysosomes, because of their very important role in white blood cells. When white blood cells phagocytize invaders, the lysosome basically is a big sack of digestive enzymes. Okay, so these enzymes are going to be produced in the rough ER, and the lysosomes, we think, pinch off basically from the Golgi body. Okay, so lots of enzymes in there. The lysosomes are going to fuse with some kind of endosome, or I should say a bacteria that's been phagocytized, and then it's all those digestive enzymes in the lysosome that are going to act to, most of the time, kill that invader, digest it, and then by exocytosis, those wastes gotten rid of. Okay, lysosomes are also important even if the cell's not phagocytotic. It's also important to kind of digest old organelles that have worn out. Okay. Mitochondria, that's the cell's powerhouse. So in eukaryotic cells, this is where most of the cell's ATP is made. It does have two membranes, an inner membrane, an outer membrane, and that electron transport chain is going to be located in that inner membrane. Okay, a couple other things that are interesting about the mitochondria. They actually have their own ribosomes, and they're of a 70S structure. So that's kind of interesting. And they also have their own DNA. Okay, and it's a small circular chromosome. Okay, so we're going to come back to the importance of that in just a second. I want to also talk about the chloroplasts. Of course, only eukaryotes that are photosynthetic are going to have chloroplasts. But if bacteria are photosynthetic, they have light harvesting pigments in their cell membrane. Of course, eukaryotes have special organelle where this happens. Also has two membranes. It's kind of similar in structure to the mitochondria. Chloroplasts also have their own DNA and also have 70S ribosomes. Okay, so primarily because of those two things, the mitochondria and the chloroplasts having their own their own DNA and having ribosomes similar to prokaryotes, that has led to a theory that was proposed by Lynn Margolis in the 1970s, and she called her theory the endosymbiotic theory. And when she first published this, she was laughed out of town. Like, people didn't take her seriously. They thought it was ridiculous. But as time went on, more and more evidence was found that supported this theory. So basically, what her theory said, it explains how eukaryotic cells evolved from prokaryotic ones. Okay, so an endosymbiont is an organism that lives inside another organism, and they're both kind of happily working together. Okay, so what she hypothesized what happened is you had some early anaerobic prokaryote that engulfed an aerobic prokaryote, and instead of killing it, they basically lived together. Okay, and this was so they became an early, basically, eukaryotic cell. Okay, so the things that support that theory, like I said, mitochondria and chloroplasts have that 70S ribosome. So mitochondria basically used to be a prokaryotic organism. Chloroplasts used to be a prokaryotic organism. And the fact that they have their own DNA. Now, today, we think that mitochondria, most of the information that it uses, it comes from the nucleus. So some of that DNA has been transferred to cell nucleus now, but DNA and mitochondria, they replicate independently of the cell. Okay, so even though they have their own DNA, now the mitochondria and chloroplasts are really dependent on the cell that they're in, but maybe it wasn't like that to begin with. Okay, and ribosomal RNA is similar in structure to prokaryotic cells. And of course, mitochondria and chloroplasts are about the same size as prokaryotic cells typically are as well. So there are a few things that maybe wouldn't line up with this theory, but for the most part, there's a lot of support that it seems to have a lot of support from most scientists now. Okay. All right, so that pretty much wraps up our chapter 3, which, whoops, the sign got chopped off a little bit there. So again, you can be working on chapter 3 of your homework. A lot of content of chapter 3. We pretty much covered most of what's in the book. Again, there's a lot of study helps and things like that in the book that are good to take a look at. Okay, we're going to spend a little bit of time here since we just talked about some eukaryotic structures. We're going to spend a little time in chapter 12 here exploring some microscopic eukaryotes. This is mostly the only time in the semester that we talk about those. So if you're particularly interested in fungi, for instance, we'll hit it a little bit now. We have a couple professors, Dr. Cleary and Dr. Thomas, that do research on eukaryotes in our microbiology department here. So I guess speak with them. We do have some upper level. There's a pathogenic fungi course, but we only really give them kind of short shift here. We've got some yeast here. We're going to talk a little bit about protozoa, not very much about algae. Here's some marine algae diatoms. So again, some examples of some of the organisms that we'll touch on a little bit. The beginning of chapter 12 does a little review of mitosis and meiosis. I didn't assign that reading. We really only need a basic idea of mitosis and meiosis when we talk about fungal reproduction. So I do, again, list the reading for every chapter, and I would just pay attention to those pages and not worry about other parts that are in the book. Okay, so we should know differences, classification between the different kinds of eukaryotes. We're going to talk a little bit about protozoa in general, and then I like to spend a little bit of time talking about malaria caused by a pathogenic protozoa, or I think they call it parasitic, actually is the correct way to say that. We will only mention algae just really briefly before we get into the fungi, spend a little bit more time there. There's a lot of special terminology with fungi that you have to understand when you especially talk about how it reproduces. So I actually have cut some material from this, some of sexual reproduction in fungi, which is sort of a relief to most students because it's mostly stuff that you've never seen before. And the book cut some of that in later editions as well. So that's kind of where we're going with chapter 12. A kind of hop back to classification here. Remember Linnaeus in his classification of all organisms, he said, well, we got plants and animals. How has that system evolved? Kind of late 1960s, Whitaker came up with his, he called bacteria Monera, and then the eukaryotes, he had the fungi, the protista, plant and animals. Today, taking into account the ribosomal RNA sequencing that Woese and Fox did, it's generally agreed that it's most correct to have many different kingdoms. And the problem is these kingdoms are not familiar to most people. So unless you're a scientist that studies some kind of protozoa, you're probably not going to be familiar with that. So they're not in big use, right, as where we stand now. But so mostly we stick with kind of these same terms that we've been using for a long time, again, because those are more familiar. But you have to recognize that there are some organisms that don't fit really nicely into calling them a fungi or a protozoa. For instance, this, let's see, this Stramenopila, those organisms are slime molds. The great potato blights in Ireland in the 1800s was caused by a slime mold. Okay, caused a lot of famine. And those organisms, they're kind of like fungi, they're kind of like algae, they're a little bit like protozoa, so they kind of share a lot of different characteristics. So, all right, so if we talk about that group protozoa, which again, is not, you know, it has some, it's not perfect, it's not a perfect term. But if we look at characteristics shared that we generally agree that protozoa share, of course, they're eukaryotes and they have one cell, so unicellular. And I mentioned this briefly, they lack cell walls, but some of them have a tenacious covering called a pellicle. Okay, I mentioned that before. Most of the time they are motile. You can find them in any body of water. That's where most protozoa live. Sometimes just moist environments. There's a few protozoa that can live there. And then there are a few protozoa that are disease causing. Like this one, these are trypanosomes, cause African sleeping sickness. Here's Giardia. And it's really interesting. It kind of looks like it has eyes, but these are just two nuclei in the cell. And this is why you don't drink even pristine water from a mountain creek, because there might be Giardia in there or Cryptosporidium that's going to give you a bad case of diarrhea. Okay, so they are mostly considered chemoheterotrophs, which is a fancy way of saying they eat stuff. Okay, they are going to get nutrients from organic sources. And like I mentioned before, most of them are free living. But there are a few examples of parasitic ones. And next time we're going to talk about one in particular, malaria, just because it is so interesting and so widespread. Okay, if you have any questions, catch me up here or catch me at my office. And I hope you all have a good weekend. Yeah, 1 second, let me stop my recording here.